Which of the following is not a cardinal sign of acute inflammation? A. Redness. B. Heat. C. Swelling. D. Itching. The correct answer is D. Itching is not considered a cardinal sign of acute inflammation. Which of the following cells is the first to arrive at the site of inflammation during the acute inflammatory response? A. Neutrophils. B. Eosinophils. C. Macrophages. D. Lymphocytes. The correct answer is A. Neutrophils are the first cells to arrive at the site of inflammation during the acute inflammatory response. Which of the following mediators is responsible for increasing vascular permeability during inflammation? A. Histamine. B. Interleukin 8. C. Tumor necrosis factor alpha. D. Prostaglandins. The correct answer is A. Histamine is a key mediator responsible for increasing vascular permeability during inflammation. In atherosclerosis, what type of white blood cell is crucial for plaque development in blood vessels? A. Neutrophils. B. Eosinophils. C. Macrophages. D. Lymphocytes. The correct answer is C. Macrophages play a crucial role in the development of plaques within blood vessels during atherosclerosis. Which of the following is a major risk factor for developing atherosclerosis? A. High-density lipoprotein cholesterol. B. Low blood pressure. C. Physical activity. D. Hypercholesterolemia. The correct answer is D. Hypercholesterolemia is a major risk factor for the development of atherosclerosis. Which of the following is a characteristic feature of chronic inflammation seen in atherosclerosis? A. Short duration. B. Predominance of neutrophils. C. Tissue destruction and repair. D. Resolution in a few days. The correct answer is C. Tissue destruction and repair are characteristic features of chronic inflammation seen in atherosclerosis. Which of the following is a key process in wound healing, especially repairing damaged blood vessels during atherosclerosis? A. Angiogenesis. B. Hemostasis. C. Fibrinolysis. D. Erythropoiesis. The correct answer is A. Angiogenesis is a key process in repairing damaged blood vessels during atherosclerosis progression and wound healing. Which of the following is a pro-inflammatory cytokine involved in the pathophysiology of atherosclerosis and contributes to endothelial dysfunction? A. Interleukin 10. B. Interleukin 6. C. Interleukin 4. D. Interleukin 12. The correct answer is B. Interleukin 6 is a pro-inflammatory cytokine involved in the pathophysiology of atherosclerosis and contributes to endothelial dysfunction. What is the primary function of foam cells in atherosclerosis? A. Promote clot formation. B. Enhance vascular tone. C. Initiate the inflammatory response. D. Engulf and store lipids. The correct answer is D. Foam cells in atherosclerosis primarily engulf and store lipids, leading to the formation of fatty streaks in blood vessels. Which of the following cell types contributes to the development of fibrous caps in atherosclerotic plaques? A. Neutrophils. B. Macrophages. C. Smooth muscle cells. D. B. Lymphocytes. The correct answer is C. Smooth muscle cells contribute to the development of fibrous caps in atherosclerotic plaques. Which of the following is a key mediator that promotes white blood cell migration to inflammation sites during the acute inflammatory response in atherosclerosis? A. Histamine. B. Leukotrienes. C. Interleukin 8. D. Tumor necrosis factor alpha. The correct answer is B. Leukotrienes are lipid mediators that play a crucial role in the recruitment and migration of white blood cells to sites of inflammation, including in atherosclerosis. Which growth factor promotes smooth muscle cell migration and proliferation, forming fibrous caps in atherosclerotic plaques? A. Insulin-like growth factor 1, or IGF-1. B. Platelet-derived growth factor, or PDGF. C. Transforming growth factor beta, or TGF-beta. D. Vascular endothelial growth factor, or VEGF. The correct answer is B. Platelet-derived growth factor, or PDGF, is a key growth factor involved in smooth muscle cell migration and proliferation. This leads to the development of fibrous caps in atherosclerotic plaques. Which of the following molecules act as chemoattractants and promote the adhesion of circulating leukocytes to endothelial cells during the initiation of inflammation in atherosclerosis? A. Selectins. B. Integrins. C. Cadherins. D. Immunoglobulins. The correct answer is A. Selectins are adhesion molecules that mediate the initial interaction between circulating leukocytes and endothelial cells during inflammation, including in atherosclerosis. Which of the following processes removes apoptotic cells and debris from phagocytic cells during the resolution of inflammation in atherosclerosis? A. Efferocytosis. B. Pyrotosis. C. Necrotosis. D. Autophagy. The correct answer is A. Efferocytosis is the process of phagocytosis of apoptotic cells and cellular debris. It's essential for resolving inflammation and promoting tissue repair, including in atherosclerosis. Which of the following enzymes is involved in breaking down the extracellular matrix and weakening the fibrous cap in advanced atherosclerotic plaques? A. Matrix metalloproteinase, or MMPs. B. Plasmin. C. Elastase. D. Hydrolase. The correct answer is A. Matrix metalloproteinase, or MMPs, are enzymes that break down the extracellular matrix. This weakens the fibrous cap in advanced atherosclerotic plaques. Which of the following lipid mediators dampen inflammation and promote its resolution in atherosclerosis? A. Resolvins. B. Leukotrienes. C. Prostaglandins. D. Thromboxanes. The correct answer is A. Resolvins are lipid mediators that help resolve inflammation and promote tissue repair, including in atherosclerosis. Which of the following processes is involved in forming a stable fibrous cap over atherosclerotic plaques, preventing rupture and thrombosis? A. Smooth muscle cell migration. B. Collagen synthesis. C. Endothelial activation. D. Macrophage infiltration. The correct answer is B. Collagen synthesis is crucial in forming a stable fibrous cap over atherosclerotic plaques, protecting against rupture and thrombosis. What is the process by which white blood cells move from blood vessels to the site of inflammation during atherosclerosis? A. Exocytosis. B. Dipedesis. C. Phagocytosis. D. Pinocytosis. The correct answer is B. Dipedesis, also known as leukocyte extravasation, is the process by which white blood cells leave the blood vessel and migrate towards the site of inflammation. Which inflammatory mediator mainly causes pain and fever in atherosclerosis? A. Histamine. B. Prostaglandins. C. Leukotrienes. D. Bradykinin. The correct answer is B. Prostaglandins are lipid mediators produced during inflammation. They induce pain and fever. Pain is induced by sensitizing pain receptors. Fever is induced by acting on the hypothalamus. What is the main function of platelets in atherosclerosis progression? A. Promote vasodilation. B. Initiate fibrinolysis. C. Enhance vascular permeability. D. Participate in thrombus formation. The correct answer is D. Platelets play a crucial role in thrombus formation at the site of atherosclerotic plaque rupture, potentially leading to myocardial infarction or stroke. Which of the following cell types secretes collagen and contributes to the formation of fibrous caps in atherosclerotic plaques? A. T lymphocytes. B. Eosinophils. C. Macrophages. D. Fibroblasts. The correct answer is D. Fibroblasts secrete collagen. They also help form fibrous caps over atherosclerotic plaques. This helps stabilize the plaques. Which of the following steps is essential for angiogenesis, a process involved in wound healing and the development of new blood vessels around atherosclerotic plaques? A. Vasodilation. B. Vascular pruning. C. Endothelial cell proliferation. D. Increased vascular permeability. The correct answer is C. Endothelial cell proliferation is crucial for angiogenesis, leading to the formation of new blood vessels that can supply oxygen and nutrients to tissues, including atherosclerotic plaques. In atherosclerosis, which type of white blood cell eats oxidized LDL particles, forming foam cells in plaques? A. Neutrophils. B. Monocytes. C. Basophils. D. Natural killer cells. The correct answer is B. Monocytes phagocytize oxidized LDL particles in the arterial wall, transforming into foam cells that contribute to the development of atherosclerotic plaques. Which of the following substances promotes the migration of smooth muscle cells into the intima of blood vessels, contributing to the formation of atherosclerotic plaques, fibrous caps? A. Nitric oxide. B. PDGF. C. Acetylcholine. D. Interleukin-6. The correct answer is B. Platelet-derived growth factor, or PDGF, is a key growth factor. It promotes the migration and proliferation of smooth muscle cells into the intima. This forms the fibrous caps of atherosclerotic plaques. What is the primary role of matrix metalloproteinase, or MMPs, in atherosclerosis progression? A. Promote fibrous cap formation. B. Stimulate vasodilation. C. Facilitate extracellular matrix degradation. D. Induce platelet aggregation. The correct answer is C. Matrix metalloproteinase, or MMPs, are enzymes that break down the extracellular matrix. This includes collagen in fibrous caps, weakening atherosclerotic plaques. Which of the following mediators is mainly responsible for smooth muscle cell migration into the blood vessel lining during atherosclerosis? A. Histamine. B. Tumor necrosis factor alpha. C. Platelet-derived growth factor. D. Interleukin-8. The correct answer is C. Platelet-derived growth factor, or PDGF, promotes smooth muscle cell migration into the intima. This leads to fibrous cap formation in atherosclerotic plaques. What is the primary function of macrophages in atherosclerosis and inflammation resolution? A. Production of collagen. B. Phagocytosis of debris and apoptotic cells. C. Activation of endothelial cells. D. Promotion of angiogenesis. The correct answer is B. Macrophages play a crucial role in phagocytizing debris, including apoptotic cells, during the resolution of inflammation in atherosclerosis. Which of the following processes is essential for the repair and remodeling of damaged blood vessels during atherosclerosis progression? A. Fibrous cap formation. B. Acute neutrophil migration. C. Angiogenesis. D. Temporary vasodilation. The correct answer is C. Angiogenesis is an essential process in repairing damaged blood vessels and promoting tissue repair during atherosclerosis progression. Which of the following molecules acts as chemoattractants and initiates the adhesion of circulating leukocytes to endothelial cells in atherosclerosis? A. Interleukin 8 or B. Prostaglandins. C. Nitric oxide. D. Bradykinin. The correct answer is A. Interleukin 8 or IL-8 acts as a chemoattractant, promoting leukocyte adhesion to endothelial cells during inflammation in atherosclerosis. What is the main function of dendritic cells in atherosclerosis? A. Formation of fibrous caps. B. Presentation of antigens to T cells. C. Phagocytosis of debris. D. Release of histamine. The correct answer is B. Dendritic cells play a crucial role in presenting antigens to T cells and modulating the immune response in atherosclerosis. Which of the following growth factors promotes endothelial cell proliferation and migration to repair damaged blood vessels in atherosclerosis? A. Tumor necrosis factor alpha. B. Vascular endothelial growth factor. C. Interleukin 6. D. Transforming growth factor beta. The correct answer is B. Vascular endothelial growth factor, or VEGF, promotes endothelial cell proliferation and migration to repair damaged blood vessels during atherosclerosis progression. What is the primary role of lymphocytes in chronic inflammation during atherosclerosis? A. Phagocytosis. B. Antigen presentation. C. Collagen production. D. Platelet activation. The correct answer is B. Lymphocytes play a role in antigen presentation and modulation of the immune response, contributing to chronic inflammation in atherosclerosis. Which cytokine activates endothelial cells and promotes adhesion molecule expression for leukocyte recruitment in atherosclerosis? A. Interferon gamma, IFN gamma. B. Interleukin 10, IL-10. C. Tumor necrosis factor alpha, TNF alpha. D. Interleukin 4, IL-4. The correct answer is C. Tumor necrosis factor alpha, TNF alpha, activates endothelial cells and induces adhesion molecule expression for leukocyte recruitment in atherosclerosis. What is the process by which new blood vessels are formed from pre-existing vessels to supply oxygen and nutrients to atherosclerotic plaques? A. Vasculogenesis. B. Extravasation. C. Angiogenesis. D. Hematopoiesis. The correct answer is C. Angiogenesis is a critical process in atherosclerosis. It involves the formation of new blood vessels from existing ones. These new vessels support plaque growth and progression. Which of the following cell types secretes cytokines and growth factors to modulate inflammation and tissue repair in atherosclerosis? A. Neutrophils. B. B lymphocytes. C. T cells. D. Macrophages. The correct answer is D. Macrophages are key immune cells in atherosclerosis. They secrete various cytokines and growth factors to regulate inflammation and tissue repair processes. Which mediator promotes smooth muscle cell migration into blood vessel intima, forming fibrous caps in atherosclerotic plaques? A. Prostaglandins. B. Tumor necrosis factor alpha, or TNF alpha. C. Transforming growth factor beta, or TGF beta. D. Interleukin 8, or. The correct answer is C. Transforming growth factor beta, or TGF beta, plays a crucial role in promoting smooth muscle cell migration into the intima. This contributes to fibrous cap formation in atherosclerosis. Which of the following processes is involved in removing apoptotic cells and cellular debris from phagocytic cells during the resolution phase of inflammation in atherosclerosis? A. Neutrophil extracellular traps formation. B. Ephrocytosis. C. Opsonization. D. Diapedesis. The correct answer is B. Ephrocytosis is the process by which phagocytic cells clear apoptotic cells and cellular debris, contributing to the resolution of inflammation in atherosclerosis. Which of the following enzymes is involved in tissue remodeling and degradation of the extracellular matrix, leading to fibrous cap weakening in advanced atherosclerotic plaques? A. Cyclooxygenase, COX. B. Lipoxygenase, LOX. C. Matrix metalloproteinase, MMPs. D. Phospholipase. The correct answer is C. Matrix metalloproteinase, or MMPs, are enzymes that help remodel tissue. They break down the extracellular matrix in advanced atherosclerotic plaques, weakening the fibrous cap. Which of the following lipid mediators is known for its anti-inflammatory properties and plays a role in resolving inflammation in atherosclerosis? A. Prostaglandins. B. Leukotrienes. C. Lipoxins. D. Thromboxans. The correct answer is C. Lipoxins are lipid mediators with anti-inflammatory properties that promote the resolution of inflammation in atherosclerosis. Which of the following molecules act as chemoattractants and promote the adhesion of circulating leukocytes to endothelial cells during the initiation of inflammation in atherosclerosis? A. Nitric oxide. B. Prostacyclin. C. Selectins. D. Integrins. The correct answer is C. Selectins are adhesion molecules that help white blood cells stick to blood vessel cells. This is important in the early stages of inflammation in atherosclerosis. Which of the following substances promotes the migration of smooth muscle cells into the inner lining of blood vessels, contributing to the formation of atherosclerotic plaques fibrous caps? A. Nitric oxide. B. Histamine. C. Platelet-derived growth factor. D. Interleukin 8. The correct answer is C. Platelet-derived growth factor, or PDGF, is a potent mitogen. It promotes the migration of smooth muscle cells into the intima. Thus contributes to fibrous cap formation in atherosclerosis. Which molecule activates endothelial cells and promotes adhesion molecule expression for leukocyte recruitment in atherosclerosis? A. Interferons. B. Toll-like receptors. C. Tumor necrosis factor alpha, or TNF-alpha. D. Interleukin Che, or. The correct answer is C. Tumor necrosis factor alpha, or TNF-alpha, activates endothelial cells and promotes adhesion molecule expression. This facilitates leukocyte recruitment during atherosclerosis. What is the primary role of matrix metalloproteinase, or MMPs, in atherosclerosis progression? A. Promoting vasoconstriction. B. Inducing apoptosis of smooth muscle cells. C. Facilitating plaque stabilization. D. Degrading extracellular matrix components. The correct answer is D. Matrix metalloproteinase, or MMPs, play a key role in degrading extracellular matrix components. This contributes to plaque instability and progression in atherosclerosis. Which process brings monocytes to inflammation in atherosclerosis, where they become macrophages and build plaque? A. Chemotaxis. B. Phagocytosis. C. Exocytosis. D. Apoptosis. The correct answer is A. Chemotaxis is the process by which cells, like monocytes, are attracted to the site of inflammation through chemical signals. Which mediator primarily helps white blood cells stick to blood vessel walls during atherosclerosis inflammation? A. Vascular endothelial growth factor, or VEGF. B. Interleukin 8, or C. P-selectin. D. Tumor necrosis factor alpha, or TNF alpha. The correct answer is C. P-selectin is an adhesion molecule that helps in the initial attachment of leukocytes to the activated endothelium during inflammation. What is the primary function of endothelial cells during the initiation of inflammation and atherosclerosis progression? A. Formation of fibrous caps. B. Chemoattractant secretion. C. Adhesion molecule expression. D. Smooth muscle cell proliferation. The correct answer is C. Endothelial cells play a crucial role in expressing adhesion molecules. These molecules help recruit leukocytes to inflammation sites. Which of the following is a critical step in resolving inflammation during atherosclerosis? It involves the phagocytic uptake of apoptotic cells and debris by specialized immune cells. A. Angiogenesis. B. Fibrosis. C. Atherosclerosis. D. Chemotaxis. The correct answer is C. Atherosclerosis is the process of phagocytosing apoptotic cells and cellular debris, contributing to inflammation resolution. What is the primary role of neutrophils in the acute inflammatory response during atherosclerosis? A. Phagocytosis of foreign particles. B. Synthesis of collagen for wound healing. C. Maintenance of chronic inflammation. D. Release of histamine for vasodilation. The correct answer is A. Neutrophils are the first responders during acute inflammation, involved in phagocytosis of pathogens and debris. Which type of white blood cell mainly becomes foam cells in atherosclerotic plaques by eating oxidized LDL particles? A. Neutrophils. B. Lymphocytes. C. Macrophages. D. Eosinophils. The correct answer is C. Macrophages phagocytize oxidized LDL particles and transform into foam cells, contributing to plaque formation. Which of the following growth factors promotes smooth muscle cell migration and proliferation in blood vessels, leading to fibrous cap formation in atherosclerotic plaques? A. Insulin-like growth factor. B. Platelet-derived growth factor. C. Fibroblast growth factor. D. Vascular endothelial growth factor. The correct answer is B. Platelet-derived growth factor, or PDGF, plays a key role in smooth muscle cell migration and proliferation in atherosclerosis. Which of the following processes is crucial for promoting wound healing and tissue repair during atherosclerosis progression? A. Angiogenesis. B. Fibrosis. C. Matrix metalloproteinase activation. D. Oxidative stress. The correct answer is A. Angiogenesis is essential for developing new blood vessels. These vessels supply oxygen and nutrients to atherosclerotic plaques, aiding in tissue repair and wound healing. Which pro-inflammatory mediator promotes oxidative stress and endothelial dysfunction in atherosclerosis, contributing to disease progression? A. Interferon gamma. B. Tumor necrosis factor alpha. C. Interleukin 10. D. Transforming growth factor beta. The correct answer is B. Tumor necrosis factor alpha, a key pro-inflammatory cytokine, plays a significant role in atherosclerosis. It does this by inducing oxidative stress and endothelial dysfunction.